can share now.
Yeah, thanks, Ivana. So I uploaded on the Zoom chat the materials for the air session. I would recommend everybody that is there with the computer to connect. And well, I can upload again the materials. We can run this script together. So here is my RStudio interface. I apologize. Well, actually, RStudio moved to a different platform. And so I found this yesterday, this POSIT platform, which is supposed to be an interactive environment. So I hope it was, for those that had not RStudio installed, was okay. Let me know if somebody is connecting a bit on the last minute. I can upload the data set and the script again. Maybe just taking also a couple of minutes to introduce myself. So I'm Serena. I am a limnologist working on plankton ecology at the INRAE, Institute INRAE in France. I work a lot with biological interaction and how the biological interaction influence the carbon cycle in pelagic environments. And well, I guess like every ecologist, I use the statistic really as a tool. I mean, I'm not a mathematician, I'm not a statistician. So yeah, as I said, it will be an interactive session. We will, well, the idea, it will be to provide an overview of some analysis that can be run on R in a quite easy way, quite straightforward to interpret. So of course I'd be happy to answer questions, but yeah, please be kind, do not get too much into the theoretical and formula things. I mean, I will try, of course, but as I say, I'm not a mathematician, I'm not a statistician, so I will try to do my best. And well, as I said, the idea is for the morning to remain quite basic and showing straightforward analysis that can be really easily applied also without too many knowledge, and also not maybe too much time to manipulate, transform data. So really some tool that can be applied easily and quickly. And this afternoon, as Elena said, we will have the interactive session, how I'm available. And so if some of you brought your own data set and would like to run some analysis and maybe getting a bit deeper also into details, of course, we will have time this afternoon. So let's start. I would recommend to download the materials and the script on a, well, easily accessible place on your computer. Well, it could be on your desktop. So you need to set the path on R of your directory. If it's on your desktop, it could be something like this. You can access easily. The good thing about RStudio is that a graphical user interface, so you don't need the command line for everything. And I think it's here also the function for opening a file on your RStudio interface. So for me, I have it on my home folder here on RStudio. So I just set the working directory. I sent yesterday the email with the packages to install. So we will use basically these 4 packages, which are quite easily also to install. Normally, it should not be particularly problematic. If you got errors or you need to see some details, I mean, you can stop me and we can have a quick look. Otherwise, again, as I said, we can have a bit closer look in details this afternoon. I will start with mainly some function from Vegan. Here is a PDF that you can download online to the Vegan tutor from Jari Oksanen. He's a very easygoing tutorial. It's like 40 pages. It's super well done, and it can really drive you through step by step different kind of analysis you can do. And so this session is a lot inspired from this tutorial. So when you work on R, as I say, you work on different packages that you need to load on your workspace. And this is the command line we will see then also after when we switch the analysis to just clear your console. But here, as you can see, we don't have any file loaded for the moment, but it can be helpful to keep in mind that you can clear your console, and sometimes when you run a lot of analysis can be helpful to get rid of a bit of different things that were already performed. And as you know, on R, you name also file and variables. So I used to call basically everything data. So it's helpful sometimes also to just clear everything and get back from the beginning. So we will start with the drivers of natural communities. We will see some multivariate analysis, as I say, mainly inspired on the vegan tutor. So the multivariate analysis basically are some analysis that we can perform to try to recognize pattern on a big series of data. And the data set that I'm using is a simplified data set from the data set of my PhD. I was studying parasite community on phytoplankton on 2 different lakes in France. And this is the data set also that I submitted to Andrea for our parasite data set. So it will, as I said, it was from my PhD, and he got helpful again. So I was happy to get back to those data. It was really a super nice travel time. Anyway, let's load our data, and we can, when we load some data, it's always helpful to have a look on this data. So as I say, these are data from 2 lakes. Here is the first lake. It's called Lake Ada, and then the second lake, it's called Lake Paven. On a seasonal survey that was run in 2007. And we have, I counted basically the parasite on different phytoplankton species, and I grouped the phytoplankton, cyanobacterial diatoms, and chlorophytes, so on the 3 main groups to, as I say, to simplify a bit things and make the tutorial not too much complicated and not too messy. We calculate different parameter infection, when the intensity, the number of the infected cell, and the prevalence of the infection. And we also have different environmental variables that were measured during the time of the survey. So we will start by analyzing first the phytoplankton community and their host. So we will split our data set. This is something that can be really easily on R. It's just enough to name your column by number. And as you can see here, we now have only the phytoplankton community parameters. What is helpful that you can do, you can immediately do a summary of your data. And so as you see here, you have all your data ordered by, you have the minimum, the quantile, the median, and the maximum. So here you can see the mean of the cell on the host, the prevalence, the intensity of the infection. Well, for example, for the diatoms, we had a minimum of 0 prevalence and a maximum of almost 24% of prevalence. Or this is just a summary of your data, but it's nice to have a first overview of your data. Another thing that I recommend to do, as you can see here, when we call the structure of your data, R doesn't recognize the data format. the data here is on the form of characters, so this is something to pay a bit of attention. We will come back also during the tutorial. A bit the formatting of the data it's not super flexible sometimes with the data format, and especially with the data, if you have it as a character, will not work. So, but it's easy. There is a function as data. You just call it, and now you can see that the data is on a data format. So just a very quick, advertisement of ggplot. Is a very nice function on R. It's super versatile, and well, Alena showed some nice graph just to show you a bit at the beginning when we start working with data. It's nice to have a first visualization. It allows to have a quick look, and as I say, ggplot, it's super versatile. I mean, you can spend entire days only in creating graph, and well, which I do not recommend, but if you have some time, why not? It can also be funny. And the community is super rich of resources and people that is also enthusiastic for giving help and advice. And I saw last week there were also Glion webinar on some advanced function of ggplot that I wanted to watch. I didn't yet, but if you have the bit of time, as I say, the opportunity, I mean, why not? I mean, it's very helpful, especially for graphical purposes. You can really do some super nice things, but let's have a look, for example, on the community of our parasite. And here, well, you can see, as Helena showed on her presentation before, you can have a nice representation of the dynamic of the host in green. Sorry, I forgot the legend. Yeah, on the green in the host, and on blue is the parasite line. So we can see immediately how actually that's true, how these dynamics are basically corresponding, and well, the parasite are there when the host are there. You can play around as I say with the axis, create a second axis. You can put you see the labels, horizontal, vertical. So again, just a little overview of some quick representation. Well, what we can do then when we have a lot of data, I would say to check for what are the potential also of the correlation of your data. You often don't need all your data. Sorry. What we can do is using the function pairs. I don't know why now is not because I have a plus here. So function pairs, as I say, which basically display your data all together in a matrix like this. Well, as you can see, is not super intuitive. So as I say, sometimes when you have a lot of data, it can be helpful to have a bit of order in that and try to figure it out which are the information that can be essential. Well, I found out a nice function to work with the pairs function that allows to visualize directly, for example, the correlation of your data. As I say, it can be important to decide also when data are autocorrelate what can be the parameter that can be more helpful for you. So this function, I found it on this paper, which is, well, the paper, it's really about working with different stressor, how to disentangle a bit the multitude of data that you have. So I put a couple of references also on this script, so feel free to have a look. And then, of course, if you need a bit of help of advice, you can always recontact me and we can have a look on this together. So we call this function, which is called panel core. So function on R in brackets like this and like this. I mean, this is really the function that you can find in the paper. So you can just simply copy in your console and run it. And then we can run again our pairs function. And we can see now that the correlation coefficients are displayed. And even by code of color, in red, the positive one, the blue, the negative one, the significance of the correlation, and in gray, the pairs that are not significant. Still, it's not super visible, so sometimes it can be helpful, as I say, to also split a bit the data and have a look group by group. So here in this case, for example, we can have a look on the pairs of the cyanobacteria, and we see that indeed the infection parameters are all correlated. So the number of infected cells correlates with the sporangia, and the prevalence as well, and also the intensity, which in any case, the intensity in these lakes was a parameter that remained relatively stable. There were not high variation. So we can confirm this also for the other groups, the diatom and the chlorophyte. So at the end, we can decide to work, for example, with the prevalence. It can be a good parameter for the infection. And so we can split our data set. And for this, there are also different packages that can be very helpful to really work a bit with data set and transform them and manipulate them in a way that you can easily then run your analysis. For example, while the select function on the dplyr package allowed really to select and pick the data that you need without getting too mad in counting the column. That, as I say, it's a very helpful and straightforward way to select column, but sometimes when you have long data set, you have, I mean, counting them is not so super easily. So you can just name the column that you want to select. For example, here we want to select the parameter that are identificative for the lake and the date. So we just select our data set and the two columns by name that we would like to select. Or also you can be more selecting with really parameters that are in the name. So if you, for example, you name all your host like we did chlorohost, diatost, cyanohost. Here you can select all the column that ends with host, and so here you have chlorohost, diatom, and so you have all your column that finish with host. And so in the same way, we select the column that are called prevalence, and then we can reconstruct the data set with the column that we just selected in here. So we have the idea of the lakes, the prevalence, and the host infection. So just a quick overview on how can be also easy to manipulate in a flexible way your data set to make it ready for the analysis is the first step, and usually is quite important. And then, I mean, when your data set is ready, we can start running some multivariate analysis. Well, I will start with the non-multidimensional scanning. It allows to represent the position of the community in a multidimensional space, and which is basically this multidimensional 2-dimension. It's just a graph, and so it allows to reduce this multiple variability of data. The point is that the MDS does not produce the statistical output. Well, what we get is a measure of stress, and the measure of the stress is it tells us how good is the fit of our data in this simplified space. So what we aim is to have stress, ideally lower than 0.05, but let's say that everything that is below 0.5 1, 2 is okay. It calculates directly the function, the distance matrix, so we can perform our analysis, and what we can see here is that we reach a stress of 0.08, so we can be quite satisfied with this analysis, and we can plot it also. So the good things about this plot function is that you can have the type T text representation, and so basically it shows your sample by name, that were named by lake and date, and here you can see also your community variables. So we see, for example, that the host, the diatom host and prevalence are on the same space. The cyanobacteria also host and prevalence are overlapping, and also the chlorophyte prevalence with their host. So we can see already that, well, something is happening between the host and their parasite, the prevalence of the infection, but well, as I said, they're not very pushed statistical output, and I don't remember if it was Yaroksanian on this vegan tutor, or I was reading some comment about the MDMS, and so the good point is that it exists, it's available, you can perform this analysis. It's very straightforward, you don't even need to transform your data, and as you can see, it's just 3 lines. You can basically visualize a bit and have a first hint on what is going on your data. But as I say, it is not very pushed, and there are other ways. Also easy, I would say, not particularly much more complicated to have a better look on your data, and especially, well, produce a bit the statistical input that will support your conclusion. So what we do now is moving to the principal component analysis. Well, is also an analysis that allows you to reduce the dimension of your data space, but it produces the principal component output of your distribution. So we will see now how to look at that and understand how the principal component can tell us a bit what's going on in the data and how to interpret them. So, well, in general, for this analysis, we need numerical variables. This is the reason why I removed the character column, date and lake. And well, as I say, it's a good practice to name your rows, your samples in a way that for you is meaningful, because with the text output, you can in any case visualize the name. So again, we use the function, and yeah, again with the numerical variable in our data set, and so we run it, and so the output of the principal component analysis is the inertia, and well, here we have well, here is the total inertia, and the principal component tells you what is the value of each axis on these components. And also we can run a B-plot and having a look. But so running a B-plot, it's helpful because, well, also here with the inertia, we could already see it, that the values are really low. And as we can see, this graph is not very clear, is not very satisfactory. So, I mean, we need to work a bit more on this data. Actually, it's not, as I say, a multidimensional scaling, but it's PCA. It's a PCA. So it needs a bit more work on the data. So what we can do, what they recommend to do, is always to have a first look on your data and how they are distributed. You can run the hist function. And then with the apply function, you apply on your data set, and here it's on the different rows. So it goes through the rows of your data, and it summarizes them, and it shows you how these data are distributed. And so as we can see here, I mean, our data are not super well distributed, and we need some transformation. So I recommend to when you want to work with a bit more push statistical analysis to have first look and transform rapidly. It's something that you can do quite quickly, and you can always have a first look with the hist function because it's quite flexible. Here we have again our data set, and for example we can test for the cubic root transformation, and we see immediately that our data distribution is getting more closer to normality with this shape. And well, I'm a big fan of the log transformation, so usually it works. As we can see, it's even better. So in general, I simply log transform my data. And so again, the numerical variables in the data set. And so as we can see it, here is our data set. Sorry, I missed some variables. So this is, as I said, sometimes when you run different lines and you call everything data, you can have a bit of progress. But yeah, here are our raw data set again. And here we log transform, and here is our data set with the prevalence and the host abundance. And now we can try to run our PCA again. And so again, the same function. And we can see now the values are a bit more consistent, and we can see that the total inertia is 69, and the first two axes account already for quite nice percentage of variability. And we can represent our PCA. We see it's really looking much better. And what we can do also with this PCA function is playing a bit with the rescaling. So it allows to more to have a better view on your graph scaling the axis. Here we set, for example, -3. And we see here the axis is going from 0 to 3 and -3. And so we can see that the vectors are better represented now. So basically, what we can do is, well, having a first look on the importance of the different axes, because on the principal component analysis, only the first two axes are represented, but maybe there are some other axes that can be meaningful and important. And here, in this case, well, we see that, well, the third axis has can be also interesting. He has some explanation on this variability, but they are really the first two axes that are mainly explaining our variability. And so we can get back to our graph and having a bit a look on the graphical interpretation, because the good things about this test and the multivariate statistic is that you can get statistical output, but also visual output. And so this tells you already some important details about your data and how to interpret them. Sorry, so you have, as we say, different vectors, and normally, well, what is considered is that the position of the vectors tells you already something. So closest the vectors are displayed in a right angle, less we consider that are correlated. So for example, there is no correlation between the chlorophytes and the cyanobacteria and the diatoms. But when the vectors are closed and going in the same direction, we can consider that are more correlated. So they account for the same correlation. And when they are really in opposite direction, we can consider there are opposite correlated. So here we can see already from this graph that, well, there is some important correlation between the host and the parasite. So what we can do then, once we have our community displayed, we can add then what are the environmental drivers and investigate how the environmental drivers account for this diversity. So we call in our data the environmental drivers, we have it here. And then what we run is the envit function that allows us to fit the environmental parameter on the PCA distribution. And what we get is an output of all the correlation of the vector with the environmental drivers and the factors, so the lake and the date. And so what we can see here is that there is no direct correlation of the environmental drivers, or the statistical value of the P are not significant, not with the lake on the date. So basically, we can see that they are not the community that we are investigating, the host and the parasite, are not in this lake directly related. They are not environmental drivers that directly explain this ordination. But so what we can do when we are interested in understanding what are the drivers of the parasite is then focused on the parasite community. And so here we run a new PCA with only the first 3 lines of our data, means the parasite, the prevalence. So we see here we have the 3 prevalence. And so we can have a look on how our principal component analysis will explain. And as we can see, the 2 axes actually explains already quite a lot of this variability. And when we plot it, we find back the same kind of pattern with, well, the different quadrant of the graph divided with the cyano prevalence, the chloro prevalence, and the diatom prevalence. And so here we can fit again our environmental drivers. What we saw it also is the trophy and the mixing regime are character drivers, and can be discarded because too many variables that, as we said before, are redundant and can be even autocorrelated. It's better to discard it. So for further analysis, I just removed these 2 parts. And so we can fit our drivers again on this parasite dynamic. But what we observe is that there is still no particularly environmental drivers that account for the parasite distribution. But when we have the host as drivers, we see that are the host that are highly influencing this distribution. And so what we can do, as I say, the principal component analysis is quite flexible and nice. Just a short overview. Sorry, I'm running out of time. I will speed up a bit. So we can extract the scores of our principal components, and so by site. So here are our lakes divided by date, but also by species. So here are all the 3 phytoplankton group and the prevalence. And what we can do, we can still use again our PAIRS function. And in this case, well, we select only the 2 axes and the 3 host abundance that can be drivers for distribution. And we can see that indeed there is a negative correlation of the cyano abundance with the PC2, the principal component axis 2. The diatom and the chlorophytes are driver for mainly for the principal component axis 1. So if we get quickly back to our distribution, what we can see is that, yeah, axis 1 is strongly correlated to the chloro and the cyano, while the axis 2 to the diatom. Okay, so this is a bit about the multivariate analysis. I will go now super quickly through the ANOVA analysis. I will try to be fast. The ANOVA. I will show it using the dataset that Elena used also to display some graph in her presentation. So in the afternoon we can have also a bit closer look on that if somebody is interested in the ANOVA analysis. So the ANOVA is the analysis of variance, so it allows to test it for significant difference between two or more independence group and to find out also what are exactly the drivers of this difference. So what we do, we clear our space now. As we see here, we have no more object in R, and we call our data set, which is well, as I said, it's from a paper that Elena wrote in 2013. Here is the reference. And so to investigate the specific specific susceptibility of the host divided by genotype to temperature across different temperature. So when we work with ANOVA, as we see here, we have some numerical variables, but we have also some variables that for us will be the explanatory variables. So these are the variables that we want to test. For example, we have, well, the abundance of our infected and uninfected host, the prevalence, and we calculate also the change rate in the treatment compared to the control of the infection and the prevalence. So when we want to work with ANOVA, we make the distinction between explanatory variables and variables that needs to be explained. So it's important to make a factor of the explanatory variables, because otherwise, R will not recognize this as a factorial variables, but especially if you have some numerical variables, it will consider like continuous variables, and so it will not work properly. So we make our factors, and again, we can explore with some ggplot. I used here the melt function to make the data set long and represent the three different graph on the same window. Again, ggplot, it's really super funny. So you can really do a lot of things with that. So here is the effect of the temperature, for example, on these three parameters, the number of infected, uninfected cells, and the prevalence, and here the effect of the genotype. So we can see immediately that, well, there was not particularly strong effect of genotype, while for the temperature we can see, especially in the extreme, as Elena showed us in the graph, there is something going on with the infection. So ANOVA can be run with one factor or multifactorial. This is also the nice things about ANOVA. It's quite flexible. What we need to do at the beginning is to remove all the rows with only 0, because he will not like it otherwise. And so the line command, it's very simple. We can just run the function HOV. And so this is our output, and the output that we get with ANOVA, well, is the degrees of freedom, and then the parameters that are interesting for us are the F value, which tells us how robust is our test. So larger is the F value, more the variation of the independent variables is real. And of course, the p-value of the F statistics. So what is important for the ANOVA, again, is, ANOVA has to be run on normally distributed data, but it's quite accepted also when the data are not perfectly normally distributed that you run an ANOVA. What is important is the homostaticity of the residuals. So what we need to do is then to test our models fit with this principle. So what we can do is plot the parameter of the ANOVA. And what is important is that here these residuals are fitting on the, well, as much as possible on the zero, and they are straight. And also here, this line, it has to be as much as possible on the straight slope, because otherwise means you have too many outliers, and this can a bit affect the reliability of your data. So again, what we can do is inspect the data. As we can observe, they are really not particularly well distributed, but yeah, if we log transform, it's getting a bit better. So what we can do is working with, again, log transform data, and then we can run again our test, and we see here it's getting better. The line is getting more straight, more closer to the zero, but still we can see here there is a group of outlier. So here the homogeneity is improved a bit, but what is recommended when your model really does not fit this assumption is to run a Kruskal-Wallis test. Here it's confirmed the statistics, the significant effect of the temperature, of the change rate of the host temperature. So it allows you also to confirm that your test is valid. What is good about ANOVA, which is not the case of the Kruskal-Wallis test, is that, well, it can be run on multi-factors, and this maybe is a part that we can skip it and see this afternoon as we are running out of time. What I just want to show you is that the good thing about the ANOVA is that then you can run a pairwise comparison, which is called the 2K test. And so when you call it, you can see that here you have the temperature test of whole range of temperature. So it tells you here, for example, that is at the highest temperature, the difference between especially 21 degrees to the other, that the test is the most significant. So the Kruskal-Wallis is not so flexible, while, as again with the ANOVA, is really possible to check for the specific which specific group are really different from one to another. And yeah, I think I can stop here, and if there are questions, maybe I can take some question, and then yeah, say, well, it's finished. It was just to show you a bit also the flexibility of the multifactorial and how to test the interaction. But again, we will have a bit of time this afternoon, so we can get a bit more into details.
Thank you.